Biochemistry and clinical pathology. Carbohydrates. Carbohydrates are important group of compounds containing carbon, hydrogen and oxygen in which the ratio of H and O is same as in water. They are represented by general molecular formula CX, H2O, Y. Figure 4.1 Flow chart of glycolytic pathway. Metabolic pathways of carbohydrate are subdivided as follows. Glycolysis, it is a oxidation of glucose or glycogen to pyruvate and lactate, Fig. 4.1. Gluconeogenesis, it is synthesis of glucose from a non-carbohydrate source like amino acid, lactate, pyruvate. Fig. 4.4. Figure 4.2 Citric acid cycle. Citric acid cycle, it is a oxidation of pyruvate to CO2 and water. Figure 4.2. Hexose monophosphate pathway, it is a oxidation of glucose by an alternative pathway to glycolysis and citric acid cycle. Figure 4.3. Figure 4.3 The HMP pathway. HMP pathway, it produces NADPH and ribose sugars required for reductive and nucleic acid synthesis. Figure 4.4 Gluconeogenesis. Proteins and amino acids, mostly amino acids, are the monomers of protein. Amino acids have an amino and a carboxylic group attached to the same alpha carbon atom. CHRCNH2, alpha amino acid. On the basis of requirements, amino acid are divided into two broad division: essential amino acid and non-essential acids. Essential amino acids, e.g., isoleucine, leucine etc., are supplied through diet as these cannot be synthesized in the body. Non-essential amino acids, e.g., alanine, glycine etc., are synthesized in the body. In the form of proteins, amino acid perform various structural, hormonal and catalytic functions essential for life. In addition to their roles in protein, these also participate in intracellular functions like nerve transmission, regulation of cell growth, biosynthesis of porphyrins, purines, pyrimidine and urea. The amino acids are derived from dietary absorption, tissue protein catabolism and synthesis in liver in body. The fate of the amino acids is shown in Fig. 4.5. Figure 4.6. Urea synthesis. Thus they are called eukaryotic. In eukaryotic organism, the ammonia in the mitochondria of hepatocytes is converted to urea via urea cycle. Fig. 4.6. Lipids, fats and fat-like substances are known as lipids and form one of the major constituents of all living plants and animal tissues. Animal fat contain mainly saturated fatty acid and glycerol, found in milk, cheese, butter, egg etc. Vegetable fat, containing mainly unsaturated fatty acid and glycerol, is found in vegetable oils. Fatty acids from acetyl-CoA is synthesized in adipose tissue and to a lesser extent in the other tissues. This process is known as lipogenesis. Two pathways exist for the synthesis of fatty acid. I. Extra-mitochondrial pathway for the synthesis of complete new fatty acid, Fig. 4.7. Figure 4.7. Synthesis of fatty acid at extra-mitochondrial system. I. Microsomal pathway of chain elongation, Fig. 4.8. Figure 4.8. Elongation of fatty acid chain. The liver normally is storage site for fat and phospholipids. It also receives from food or from the fat depots a continuous supply of fat which is dealt by the process of B-oxidation, Fig. 4.9. Enzymes. Enzymes are soluble in colloidal organic catalysts formed by living cells. They are protein-specific in action to catalyze the chemical reaction. The enzyme have following properties. 1. Enzymes are product of living cells. E. They are more powerful than inorganic catalyst. E. They have capability of initiating a reaction and also accelerating it to many times. Figure 4.9 Beta oxidation of fatty acids. IV. The enzymatic action does not depend upon the quantity. V. Enzymes can induce reversible reaction. For example, enzyme glycogenase in liver converts glucose into glycogen for storage and again, when required, glycogenase reconverts glycogen into glucose. V. They are colloidal in nature with large molecular weight and hence mostly non-diffusible. V. Enzymes are destroyed by moist heat above 70 degrees Celsius. Action is reattited at lower temperature. Optimum activity lies between 25 degrees to 45 degrees. V. Enzymes are specific in their activity. X. Some enzymes require a counter-enzymes, usually some inorganic ion or an activator for proper enzymatic action. Coenzymes. Many enzymatic reactions are catalyzed only in the presence of specific non-protein organic molecules, called the coenzymes. They are regarded as co-substrate or prosthetic groups of enzymes. Their functions is usually to accept atoms or groups from a substrate and to transfer them to other molecules. Some coenzymes along with their activity are enlisted in Table 41. They are less specific than our enzymes, hence same coenzymes can act as such in a number of different reactions. Table 4.1 Common coenzymes for enzymatic reactions. Coenzymes for oxidoreductases: nicotinamide adenine dinucleotide, NAD. Action: hydrogen transfer vitamins nicotinamide. Coenzymes: nicotinamide adenine dinucleotide phosphate, NADP. Action: hydrogen transfer vitamins nicotinamide. Coenzymes: flavin adenine dinucleotide, FAD. Action: hydrogen transfer vitamins riboflavin. Coenzymes: flavin mononucleotide, FMN. Action: hydrogen transfer vitamins riboflavin. Coenzymes for transferases: adenine triphosphate, ATP. Action: phosphate group transfer vitamins. Coenzymes: thiamine pyrophosphate, TPP. Action: active acetaldehyde transfer vitamins thiamine. Coenzymes A. Action: acyl group transfer vitamins thiamine. Coenzymes for isomerases and lyase anodine diphosphate, UDP. Action: sugar isomerization vitamins pyridoxine. Coenzymes: pyridoxal phosphate. Action: decarboxylation vitamins E. Isoenzymes. There are different forms of the same enzymatic activity found in the same cell or tissue. They usually differ each other chemically, immunologically and electrophoretically. For example, lactate dehydrogenase, of which 5 different forms are identified electrophoretically. These enzymes catalyze a conversion of pyruvate to lactic acid. These enzymes, protein, consist of 4 subunits and that 2 very different polypeptides are involved. One type H form, heart form, contain all of one polypeptide, H. The M or muscle form contain the other polypeptide.
M4. The other 3 types consist of other 3 possible combination of the H and M polypeptides to form a tetrameric structure, MH. MH2, MH3. The M form is found in anaerobic tissues and produces lactate at rapid rate in the presence of high concentration of pyruvate. The H form predominates in aerobic tissues and is inhibited by pyruvate. Its function is probably to oxidize lactate to pyruvate, which can be utilized aerobically rather than to convert pyruvate to lactate. Water and mineral metabolism. Water is the major constituent of the body. It is distributed in 2 compartments in the body. 1. Intracellular water. Water present within the cell. 2. Extracellular water. Water present outside the cell. It is further divided into the following compartment. 1. Plasma. It is the extracellular fluid of the blood. It comprises 7.5% of the body weight. E. Interstitial fluid. It represents 20% of the body weight. E. Dense connective tissue. It include the water content in the bones and cartilages. Biological importance. 1. Water is essential component of the cell to provide the aqueous media for chemical reaction. 2. It can absorb or gives heat due to high specific heat without any appreciable change in temperature. 3. It has a very high latent heat, hence it provides a mechanism of heat regulation through sweating. 4. Water provide the fluidity to the blood. 5. Water provide the lubricant action on rubbing surfaces such as joints, conjunctiva and peritoneum to prevent friction. Water metabolism is regulated by following means. I. Concentration of the solutes. Osmotic forces are the principal mechanism for controlling the amount of water in the body. These are maintained by concentration of solutes in the body water. The solutes are of these types. A. Organic compounds of small molecular size like glucose, amino acid if present in high concentration, can help in retaining water due to osmotic effect. B. Organic substance of large molecular size, protein, can also affect the transfer of fluids from one compartment to the other. C. The inorganic electrolytes like sodium chloride and KCl are the most important both in distribution and retention of the body water. E. Water metabolism is also regulated by the hormones like vasopressin, aldosterone. Vasopressin, antidiuretic hormone, enhances the reabsorption of water and so helps in retention of water in the body. Aldosterone secreted by adrenal gland also increases the reabsorption of sodium and chloride by the renal tubule and hence water is also retained. Kinin, produced in the kidney, counteract the action of antidiuretic hormone and hence increase salt and water excretion.
Mineral metabolism. Mineral elements are supplied through the diet. Soil minerals elements like calcium, sodium, potassium, phosphorus, chlorine, are essential for life processes and present in more amount. Hence these are called principal elements, macronutrients, table 4.2. Other elements like iron, iodine, copper, zinc, manganese etc. are present in small amounts, so these are called trace elements, table 4.3. Table 4.2. Details of macronutrients. Elements: calcium. Sources: dairy products, beans, leafy vegetables. Functions: constituent of bones. Children: teeth, regulation of nerve and muscle function. Deficiency disease or symptoms: children, rickets, adults, osteomalacia. May contribute to osteoporosis. Toxicity disease or symptoms occurs with excess absorption due to hypervitaminosis D or hypercalcemia due to hyperparathyroidism, or idiopathic hypercalcemia. Elements: phosphorus. Sources: phosphate food additives. Functions: constituent of bones, teeth, ATP, phosphorylated metabolic intermediates. Nucleic acids. Deficiency disease or symptoms: children, rickets. Toxicity disease or symptoms: adults, osteomalacia. Elements: sodium. Sources: table salt. Salt added to prepared food. Functions: principal cation in extracellular fluid, regulates plasma volume, acid-base balance, nerve and muscle functions, Na, K at pace. Deficiency disease or symptoms unknown on normal diet, secondary to injury or illness. Toxicity disease or symptoms: hypertension in susceptible individuals. Elements: potassium. Sources: vegetables, fruit, nuts. Functions: principal cation in intracellular fluid, regulation of nerve and muscle function. Na, KATPS. Deficiency disease or symptoms: cardiac arrest, small bowel ulcers. Toxicity disease or symptoms occurs secondary to illness. Injury, or diuretic therapy, muscular weakness. Paralysis, mental, confusion. Elements: chloride. Sources: table salt. Functions: fluid and electrolyte balance, gastric fluid chloride, shift in HCO transport in erythrocytes. Deficiency disease or symptoms secondary to vomiting, diuretic therapy, renal disease. Toxicity disease or symptoms: elements: magnesium. Sources: leafy green vegetables, containing chlorophyll. Functions: constituent of bones, teeth, enzyme activator, kinases, etc. Deficiency disease or symptoms secondary to malabsorption or diarrhea, alcoholism. Toxicity disease or symptoms depressed deep tendon reflexes and respiration. Table 4.3. Details of essential micronutrients, trace elements. Elements: chromium. Sources: meat, liver, brewer's yeast. Whole grains, nuts, cheese. Functions: a constituent of glucose tolerance factor, which binds to and potentiates insulin. Deficiency disease or symptoms: impaired glucose tolerance, secondary to parenteral nutrition. Toxicity disease or symptoms: elements: cobalt. Sources: foods of animals' origin. Functions: required only as a constituent of vitamin B12. Deficiency disease or symptoms: vitamin B12 deficiency. Toxicity disease or symptoms: anemia. Elements: copper. Sources: liver. Functions: constituent of oxidase enzymes, cytochrome oxidase, etc., cystosolic superoxide dismutase, role in iron absorption. Deficiency disease or symptoms: anemia, hyperchromic, microcytic, secondary to malnutrition. Menkes syndrome. Sources: toxicity disease or symptoms: rare, secondary to Wilson's disease. Elements: iodine. Sources: iodized salt, seafood. Functions: constituents of thyroxine, triiodithyronine. Deficiency disease or symptoms: children, creatinism, adults, goiter and hypothyroidism, myxedema. Toxicity disease or symptoms: thyrotoxicosis. Goiter. Elements: iron. Sources: red meat, liver, eggs, iron cookware. Functions: constituent of heme enzymes, hemoglobin, cytochromes, etc. Deficiency disease or symptoms: anemia, hyperchromic microcytic. Toxicity disease or symptoms: siderosis, hereditary hemochromatosis. Elements: manganese. Functions: cofactor of hydrolase, decarboxylase, and transferase enzymes. Glycoprotein and proteoglycan synthesis. Toxicity. Toxicity. Deficiency. Mitochondrial superoxide dismutase. Deficiency disease or symptoms unknown in humans. Disease or symptoms: inhalation poisoning produces psychotic symptoms and Parkinsonism. Elements: molybdenum. Sources: functions: constituent of oxidase enzymes, xanthine oxidase. Deficiency disease or symptoms secondary to parenteral nutrition. Disease or symptoms: elements: selenium. Sources: plants, but varies with soil content, meat. Functions: constituent of glutathione peroxidase. Disease or symptoms: marginal deficiency when soil content is low, secondary to parenteral nutrition, protein energy malnutrition. Toxicity disease or symptoms: megadose E supplementation induces hair loss, dermatitism and irritability. Toxicity. Elements: silicone. Sources: plant foods. Functions: role in calcification of bone and in glycosaminoglycan metabolism in cartilage and connective tissue. Deficiency disease or symptoms: impairment of normal growth. Disease or symptoms: silicosis due to long-term inhalation of silica. Dust. Elements: zinc. Sources: functions: cofactor of many enzymes, lactate dehydrogenase, alkaline phosphatase, carbonic anhydrase, etc. Deficiency disease or symptoms: hypergonadism, growth failure, impaired wound healing, decreased taste and smell acuity, secondary to acrodermatitis enteropathica, parenteral nutrition. Toxicity disease or symptoms: elements: fluoride. Sources: drinking water. Functions: increase hardness of bones and teeth. Deficiency disease or symptoms: dental caries, osteoporosis. Toxicity disease or symptoms: dental fluorosis. Vitamins. Vitamins are organic catalysts. They are found in minute quantity. They are of no caloric value, but still they are vital to physiological processes of the human body. They are classified on the basis of their solubility. Fig. 4.10. Vitamin D divided into two types. 1. Fat-soluble vitamins. 2. Water-soluble vitamins. 1. Fat-soluble are 4 types. I. Vitamin A, E, vitamin D, E, vitamin E, IV, vitamin K2. Water-soluble vitamins are 2 types. I. Non-B complex vitamin C, EB complex vitamin B complex. EB complex vitamin B complex divided into 3 types. 1. Energy releasing. I. Vitamin E, vitamin B2. 2. Hemopoietic. I. Folic acid. E. Vitamin B2. 3. Others. I. Pantothenic acid. E. Biotin, niacin. Fig. 4.10 Classification of vitamins.